Hello everyone, welcome to Mohit Agriculture Tutorials. My name is Mohit Gupta and today's video is going to be very interesting one, right? So today's video is on what topic? The main question arises. Now which topic is taught today is going to be taught today. So the topic naming vascular traffic inside the cell, right? Cellular vascular traffic. We are going to talk about how does vascular traffic works inside the cell. How does endocytic pathway means how are vesicles that have a kind of, we can say, modified elements inside it, that may be protein, carbohydrates or lipids. How does they carry to a position where they should be sorted to, right? Or where they should be landed, or where they should be delivered, right? So delivery process of endoplasmic reticulum is divided of many variants, right? We do not have any consideration about how does this process happen, but certain laboratory techniques that I have already told you earlier also, that certain laboratory techniques are there which are being illustrated well, and they are so well illustrated that cannot be, we cannot say it a false statement at all. That's why we say them as laws, right? Now herein, in this video, we are going to talk about how does endocytic pathway, or we can say how does the cellular mechanism of transfer of protein from one area to the another area. Say what? Say we already know that Golgi apparatus and endoplasmic reticulum work together in delivering the elements to some place, some designated place. Now let's think of a kind of simple query over here. Say the endoplasmic reticulum is carrying a protein that is needed at plasma membrane, that is an integral protein. Now how does the endoplasmic reticulum transfer that protein to particular area where it is designed to, right? To the place where it should be landed. How can endoplasmic reticulum know about that, and how can it deliver at the position where it want to deliver to, right? So this answer of this question was a bit tricky, but we made it easy. We are going to talk about how does endoplasmic reticulum and Golgi body and many more, lysosome and all those bodies working all together in vascular traffic, how does they mediate the process of what, say, vascular traffic, the transfer of any of the element from one part of cell to the another, right? Say now we are going to next image. Now very first let's understand what we have shown here only. So here we have shown you can see this is a endoplasmic reticulum. Now this is a Golgi apparatus, right? Endoplasmic reticulum is beneath it, right? Let's show that also. Let's show you where is endoplasmic reticulum, right? Okay, so herein we can see in the diagram, right? So this box are coming in front of diagram, that doesn't matter, we are showing a correct diagram at correct position. So you can see herein the nucleus is being surrounded by a wall known as endoplasmic reticulum, which are inhaled with proteins or particular, we can say, elements that are to be delivered to particular position. Now if you see here, this is a rough endoplasmic reticulum. It is very clear shown over here. This is a rough endoplasmic reticulum, and if you can see here, this is endoplasmic reticulum lumen, right? From the endoplasmic reticulum lumen, if you can see here, the buds are pinching out, that we call vesicle. Vesicles are continuously pinching out from the endoplasmic reticulum and are delivered to Golgi apparatus, right? Now Golgi apparatus we have already discussed on our channel, so we do not consider it to be discussed again. Structure is not discussed, but certain properties of Golgi is to be discussed over here. If you see herein, in this picture, the Golgi is just a type of membranous, we can say, membranous sac-like structure which enables to deliver the vesicle at proper appropriate position wherein it should be delivered to, right? Now the main question arises is that how can Golgi be so appropriate to deliver at particular position wherein the vesicle are to be delivered? How it is possible, man? Means we have designed certain vesicle that are to be delivered at some position, certain position, but how can we know that at what position it is to be delivered, and how we can actually, we can say, negotiate, or we can say what, how we can actually perform the machinery without knowing any address, right? So there is a point of view. The point is that some address is labeled in these vesicles. That address is to be known. Now how does that address to be assigned? What is the address? So herein, if you can see, we have shown we can say wall of this Golgi, right? So if you see herein, the Golgi apparatus is pinching out the vesicle from its, we can say what, say it is a donor. We cannot consider it as a Golgi. It may be endoplasmic reticulum also, but the thing is, the thing to consider over here is they're showing certain ligand and receptor binding, right? So this cup type of needle and cup joined together with some balls in it, so we call these balls as ligand and this cup type of structure as a receptor. So what are these ligands and receptor? Actually this ligand and receptor has particular specific work to do. These actually decide, these particles actually decide to where to transfer certain vesicle to, right? At what position should it be transferred? Say we have to transfer it in plasma membrane. How can we know without any particular correspondence by any organelle, or how can we know by any correspondence of any, we can say, address slip we are not having? So how can we deliver at a particular position where we need to deliver to, right? So to deliver it, we have shown certain receptor and ligand binding that actually is producing a signal to where it should be delivered to, but this signal is not of particular cause, because if you see here that to deliver from one lumen to the another lumen of maybe endoplasmic reticulum, it is creating signals, but these signals are not designated to be carried. Now vesicle are to be carried at another point of the cell, say at the very end of the cell. Now to carry it at the very end of the cell, vesicle should have certain modified type of, we can say, modified type of what? Signals, right? Address slip it should have with it. Now from where will it get that address slip? So here we can see that Golgi is actually delivering these all vesicle to the membrane that we call at plasma, right? Here we can see, but how it is designated, because you see herein there are some position where these vesicle are to be delivered, right? These are containing somewhat nutrients, I think so, and these nutrients are delivered at plasma, are designated position wherein you can see the loop there is no such loop in the plasma membrane. They are just showing it a part type of describing how can the vesicle be delivered to the position, right? So how can one signal designate a position? How can a signal design a position where the vesicle is to be delivered? Now this answer is really very big question. Actually this question is very big question, but answer of this question is going to be little interesting type, right? We'll see in a moment what is the answer of. Okay, so answer of the question comes from signal hypothesis. Now what is signal hypothesis? How does it work? What are all the fundamentals of signal hypothesis? So herein you can see the images of signal hypothesis. How does it work is a bit typical to understand, but here a thing to understand is that here are the porins, we call it plug, right? Like you have a electric cable in your house wherein you have to switch the plug in and you have to switch on the plug, right? So it is a kind of plug you can see over here. There are certain receptors which usually binds to somewhat message. So if you see here is SRP, right? So this is somewhat a protein, right, that is used in signal hypothesis, right? So signal regulatory proteins these are, and these are just we can say what there is a receptor to which the ligand is to be bound to, right? So this SRP, as soon as ribosome, you can see here in the ribosome is, and the ribosome tends to come at the position where the plug is, there where the translocon is, and there only it can create a signal, right? Wherein the SRP, the signal peptide that is designated for a signal, is being inserted to the receptor, and the ribosome are going to sit at what say porin area in the translocon, and from there the protein is to be created. Now herein, if you see what protein is created, there is a BIP chaperone. Now this BIP chaperone is having one of the important function in molecular biology, right? Because these chaperone are the one that are designated for mutability. To understand what mutation is, you can see my channel has some videos on mutation, not all, but in human molecular genetics we are going to bring videos, animated videos on what say mutations, right? Now here if you see that ribosome is sitting on the wall of endoplasmic reticulum, wherein the translocon is there, and it created a signal hypothesis, and by a signal hypothesis by SRP, if you see here only we have shown the SRP. So as SRP insert to the receptor area, the receptor creates a signal, and the signal hypothesis creates some programmed, we can say infrastructure that we call protein. Once protein is created by transcription translation of that mRNA, now once protein is created, we call it BIP chaperone. Now keep this fundamental concept of BIP chaperone at side now, because we are not trying to understand BIP right now. We are trying to understand what is signal hypothesis. So signal hypothesis is very simple. We say that there are some designated signals in an endoplasmic reticulum membrane to let it to create some proteins that are to be sorted out and transported from one part of the cell to the another, right? So this is one of the case. Let's go to another case. Okay, so again one of the very basic and important diagram that shows here all the things, the signal hypothesis. So if you see here in, this is the membrane of endoplasmic reticulum. There is a designated, we can say, protein, membrane protein, which is creating a signal. If you see here, ribosome binds to this protein, and this protein actually serves as a mediator for what? Say a protein is assigned, or we can say a protein is generated by the ribosome. Now it is to be created somewhat impact is to be created that the protein should go inside the lumen, not out. So to just generate we can say passage of protein from the ribosome to the lumen of endoplasmic reticulum, these we can say membrane proteins are serving as the channel, right? A kind of, we can say, to reduce the uses of ATPs, these ribosomes are actually creating a gateway to give the protein a passage from the translocon unit to the lumen of endoplasmic reticulum, right? So this was also one of the important diagram. So here is shown one more diagram that is very important, but to understand it we must know the fundamentals of how does these all things work together, right? So altogether these fundamental diagrams are assigned to give us a knowledge, we can say a quick preface of what is the transport of vesicle, right? How does vesicle transport the designated, we can say, protein or anything to particular position where it is designed to, right? So if you see herein, the vesicles are actually, we can say, budded out of Golgi, and they are carrying some vesicle membrane, and they are carrying some, we can say, exterior proteins type of lipid. You can see here lipid tail is there, and it is designed in such a way that it is to be transferred in the at the position wherein the plasma membrane is curved somewhat, right? Now the big question arises is that how can be these organelles so accurate? What is the cause of accuracy? Is there anything inbuilt in these organelles that they are so much accurate, or there is something also working continuously with these organelles to serve as, right? Now we'll learn these part of things within a while. Let's go to the another diagrams, right? Okay, so now here comes the answer of what we are actually trying to get off, right? So here you can see there is a cell, right? This is not a cell actually, this is endoplasmic reticulum, lumen, right? I just mistaken said that it is cell, right? So it is the lumen of endoplasmic reticulum, wherein you can see here the protein is generated by this ribosome, right? Now things to remember is that ribosome once generates protein, it is to be modified inside the lumen of endoplasmic reticulum due to we have to sort out the particular protein that is to be assigned, or that is to be, we can say, is thought to be delivered somewhere in the cell. How can one know, how can an organelle know where to deliver, what vesicles should the cargo loaded to, that it is to be delivered at correct position? So herein when we see that this ribosome creates a protein that is somewhat modified. If you can see, the N terminal is somewhat modified. So what kind of modification does it have? Now this modification is said as polysaccharide modifications on, say what, N terminal of the amino acid side chain, right? So if you see herein, there are various amino acids, asparagine, serine, threonine. Now these amino acids are designated at N chain, N terminal. If you see herein, that this is the carboxylic terminal and this is N terminal. So carboxylic terminal is very right near to ribosome, and N terminal is very right near to the lumen of endoplasmic reticulum. So if you want to have an idea how does the protein is delivered at a particular correct position, then these sugars are added. Now let's call this sugar is N-acetyl glycosamine, NAG. After that mannose, this is a triangular shape, that's why mannose, glucose, and at some part of view, right, not every time, dolicol is also added, right, dolicol phosphate. Now herein they are showing that dolicol phosphate is already inbuilt in the membrane of endoplasmic reticulum. Now this diagram is very up clear, right? So they are showing very clarity about how the protein is assigned to be delivered at particular position, but this is not so correct. This signaling hypothesis says that these mannose and dolicol phosphate, glucose, and acetyl glycosamine, whatever is added, whichever sugar is added, it is only assigned to be loaded up till Golgi apparatus. Means this cargo is, we can say, designated to be only, we can say, unloaded at Golgi apparatus. It will not go somewhere else. It will not go directly to the lysosome via Golgi apparatus. Now there are some more modification in the pathway of this type of process, right? We'll learn what is the modification process, right? Okay, so a very accurate diagram we got at this position. We need such a diagram only. So herein you can see that this is the endoplasmic reticulum wall, right? Now the problem creates when the endoplasmic reticulum ribosome actually creates certain protein that are misfolded that are mutated proteins. Now once these proteins are mutated, they're not to be sorted out of the cell. If they are sorted out or carried, or the cargo is unloaded at certain position where it is designated to, then it may create something that may be deleterious, right? That must be destructive, right? So how can these protein be sorted out, and how can these protein be, we can say, destructed inside the cell only? Now there is a kind of signal hypothesis. There is one more kind of signal hypothesis. Now at the very first diagram we have shown that the BIP is generated inside the lumen of endoplasmic reticulum. So here we can see there is a BIP. Now we can see that there are somewhat receptors, or we can say a type of protein. We call these protein, we do not consider any name to this protein right now, but proteins are important for our use too, right? So we say this protein has a PERK protein, right? So these PERK proteins are really very important. So here we can see that at the very first step, right? So let's see this properly. So at the very first step, the BIP binds. BIP is a kind of protein, right? So it binds to these complexes held in the lumen, not in the lumen, in the wall of endoplasmic reticulum. These are the inactive sensors, we can say so, right? Now we can say these are type of receptors that are thought to be bound to any ligand to get active, right? So herein we can see, as soon as the BIP binds to these receptors, we can say what? These receptor PERK receptors dimerizes. Now once it is dimerizing, it will create somewhat phosphate pump. So phosphate shuttle is there. Now there is one kind of protein that will use to come and settle at the these dimerized proteins, right? Receptors. Now once this EIF2A alpha protein, as soon as it comes and settle at this position, it will create somewhat phosphate shuttle and phosphorylate these protein. Now once this protein is phosphorylated, if you can see herein, it is just carried to the ribosome, and wherein ribosome creates a signal. Now this signal is created by mRNA, that is transcribed by the nuclear DNA. As this protein has been transcribed at the step 5A, you can see herein the protein are capable of elevating endoplasmic reticulum stress, and as the endoplasmic reticulum stress is elevated, so we can actually not destroy, that protein will be again sent to, because this process, the whole process is very much calorie wasting, right? ATP using process. So to decide a proper fundamental process, the proper treatment should be given to the protein. So it is again taken towards the endoplasmic reticulum lumen. Again enzymes work over there to modify the byproduct, right? The protein that was unfolded. Okay, let's go to another